So let's get started. Today we're talking about plant viruses, and don't tell any of my colleagues down at Oregon State that we're covering plant viruses in one lecture, because they would probably run me up the flagpole. Really completely unfair to this really massive and fascinating group of viruses. And maybe even to do complete injustice, if we get done early, I'm going to go back and talk about some of the pandemic flu stuff at the end. Yeah?
Just to be clear, we have a test on Wednesday, right?
That would be my understanding, yes. What if we had it Friday instead? That's not going to work terribly well because I'm not even going to be on campus on Thursday, so I need to have it done way earlier than that. So what's the point? So,
So since science is done in teams,
yes. We're doing clickers as teams already. That was a good try. I like. Yep. So, any more questions about the exam? So there will be a review session on Monday, which will just cover old stuff, but there will be clicker questions from today. Okay. Any other questions, comments, worries? It will just be on the stuff that we've covered in lecture, so not the extra stuff at the end of lectures that we haven't gotten to. And it's nice because it's all the RNA viruses. See, it's just a nice focused unit. It's another reason not to put it off until Friday, because then we'd have these nasty DNA viruses in there too. Ugh. We don't want that. And putting it off a day would mean there would actually be more material in the exam, which we might not want either. No? Okay, so speaking of group work, let's actually do a group question. How many genome segments are there in the H1N1 swine flu? 1, 2, 4, 6, or 8. And again, this is where you can work as a group. Science is done in groups. Ten, 5, 2, 1, 0. What flavor of influenza, A, B, or C, is H1N1? A. How many genome segments does influenza A have? Oops. It is 8. Yes. So, oh, come on. Click this thing too many times. We'll talk more about other genome segments for some of the other viruses, and particularly for the CucumberMosaic virus. This is really fascinating. It's got 3 segments, but they're packaged in 3 different virions, which is really bizarre. And 4 segments, in fact, even in some cases. So, without further ado, the decision on what will be used in next year's influenza vaccine depends on bird flu, the weather, the proportion of influenza A and B, how much genome reassortment took place, or what influenza was present on the other side of the world. Group. I like group. Group, good. F. F. It's always F. F. So who else got Obama'd on the way in today? Yes, we all got Obama. That's why we're late. No?
It was annoying because everyone in front of me, there's no traffic going eastbound. There's hardly anybody. But the people that were there were driving 35 so that they could see the convoy.
So everybody today should get an Obama point for just being here. That's it. But what does he care about plant viruses? Yeah, and they trade agreements and those kinds of things.
Not quite free enough.
Three, 2, 1, 0. Yes, it's what's happening on the other side of the world, southern hemisphere. So can I just leave now because the weather is so wonderful and just we take off? And then you guys just listen to the lecture online and whatever it happens? Yes, sounds wonderful. I like to think there's actually some value added to me being here and you being here. Again, I like to think that.
You're adding to each lecture a little each year, right?
Sometimes a little, sometimes a lot. Yeah. What am I adding to each lecture each year? Which unfortunately means that might actually be on an exam. Yuck. So, plant viruses. This particular plant virus here is CCMV, cowpea chlorotic mottle virus that we'll talk about a little bit more. I really like this virus for a couple of reasons. One is, of course, it's this beautiful T equals 3 quasi-equivalent icosahedral structure, which everyone's really happy about. But more importantly is that it has this really fascinating conformational change that takes place. Basically, sort of expands. I don't know how many of you have seen these kids' toys with the wonderful expansion, contraction kind of thing. It's exactly what this virus does. It's so cool. So we'll talk a little bit more about that later on. The professor who I worked with during my postdoc worked on this and has done some really cool nanotechnology kinds of stuff with this. So yeah, we'll talk about RNA viruses. Again, plant viruses, totally not doing them justice in this period of time. The bromoviruses or the Mosaic viruses, the CucumberMosaic viruses, particularly that's what the chapter is about and one of the ones that we know the most about. We mentioned already that these guys have segmented genomes, but unlike the segmented genomes in flu, which are all in one virion, they're split up into multiple virions, which is just crazy that this is how evolution has worked for these things. One of the really critical things about these plant viruses, and probably why they can evolve to have these segmented genomes, is it's actually really hard to infect a plant. If you think about a plant, it's sitting in one place for most of its life, unless you have distribution of seeds and that kind of thing. So it's evolved to be really good at protecting itself, not surprisingly, because it can't run away. So once you have a virus infection gets into part of a plant, getting to other parts of the plant is really kind of tough. And so all plant viruses have these so-called movement proteins where they can get from one part of the plant to the other. So this is really quite different compared to any of the other viruses really we'll be talking about right now. We're all mushy and easy to get around in, but plants are definitely not that case. And then at the end we'll talk a little bit about TMV, and again, a little bit because this is the book about TMV, actually one of the books, and only from 1930 to 1965, so 35 years of TMV. Why is TMV so exciting, among other reasons? First virus ever discovered. So, and that was well before 1930. So again, our key concepts, most of the plant viruses, interestingly enough, are single-stranded RNA viruses, positive sense. Very few negative strand, very few double-stranded, few single-stranded DNA, very few double-stranded DNA viruses. So quite why that is still kind of an open question. Almost all of them have segmented genomes, Again, not all of them TMVs, and of course an exception to that. Talk a little bit about plants and why plants are different, mostly again because they can't run away. Conformational changes in the virion. We looked at that already with CCMV. We'll talk about it a little bit more. What happens to the host on these virus infections. And then I wanted to tell a really cool story that actually is not really a plant virus. It's a fungal virus, but it has some really fascinating effects on what happens to the plant. And then we'll talk at the end about some helical structures, particularly for TMV. We sort of skimmed over them really quickly when we talked about symmetry at the very beginning of the course. So the title of the chapter is CucumberMosaic virus. We'll talk about that, but we'll also, again, talk about a number of these other ones. Where did it come from? Again, this is mostly the viral disease. The structure, which is this beautiful, again, T=3 quasi-symmetric structure. How does it get in? This is probably one of these things that I probably should have highlighted in red because it's clearly very different relative to all the other viruses we've talked about so far. Replication is actually not too terribly different than a lot of the other RNA viruses we've talked about, single-stranded RNA viruses, and packaging. Some of the big differences, again, we put it in the second column over here, are satellites. Satellite RNAs, they're not really satellite viruses because they don't have any virions. They're just these RNAs that are floating around together with the virus. They need a virus in order to be able to replicate to cause disease, and they can modulate that disease. They don't even code for usually any open reading frames. And so how this little RNA could have so much influence is really pretty impressive. Talk a little bit more about CCMV and some of the applications here of that conformational change, some hot viruses, because hot viruses are cool, and tobacco mosaic virus. So this is a reminder. We're talking about positive strand, single-stranded RNA viruses, which of course can also serve as messenger RNAs. So all you need to do is get that RNA inside the cell. Once that RNA is inside the cell, as long as it looks okay, and what looks okay in terms of a messenger RNA, what do you need? Caps, and to some extent tails, but most importantly caps to get your translation, this little double blob here, the ribosome, for going on here. So these are single-stranded RNA viruses. The vast majority of viruses that people have studied in plants are these single-stranded RNA viruses. What does it look like? This is a cucumber that's been infected by CucumberMosaic virus. A mosaic phenotype just basically means that you've got bits which are green and bits which are not green. If you go back and look at Beijerinck's original paper in 1898, if I remember correctly, he talks about albinism. And that's because lacking the coloration here. So this is a really classic kind of phenotype for your disease. CucumberMosaic virus was originally discovered in 1916, and this is sort of the naming that all the plant people use for their viruses. So what's the host? Cucumber. What's the phenotype? Mosaic. So cucumoviruses. Again, I like their picoRNA viruses, they picorna viruses. Potty viruses, so be a potato yellow virus. So that's where they come up with all these names. And I didn't bring the virus taxonomy book, but it's just filled with all of these things. And as I mentioned, CCMV, cowpea chlorotic model virus. So it tells you what it infects and what kinds of phenotype you actually get. CucumberMosaic virus is really interesting because it's probably, at least as far as species are concerned, the virus with the widest host range that has ever been found. Last count, something like 1,200 different species of plants can be infected by this CucumberMosaic virus, which gets back to this naming convention. Okay, it's CucumberMosaic virus, but it can infect tobacco. Okay, wait a minute. So it's the CucumberMosaic virus infecting tobacco. So the nomenclature here, it's the first one they discovered. Yes, it was in cucumber, but infects all kinds of different things. So unfortunate, yeah, it makes sense for your first one, but if you have a wide host range, then it's not particularly good. Now, the reasoning behind the whole wide host range is still pretty up in the air. Why should you have something with such a wide host range? Because most of the viruses we've talked about for this class and in general, have very specific host ranges, very few organisms, or even in some cases, cell types that they can infect. Here, it's very different. And a lot of that has to do with the mechanism of transmission, how you actually get from one place to another. So again, hopefully we're not beating this too much over the head. T equals 3, quasi-equivalent icosahedral structure. Quasi-equivalence because it's one and only one coat protein, which is put together either in pentamers, so your 5-fold symmetry here, or hexamers, the 6-fold symmetry right here. So if you're going to do our T numbers, start here. H is equal to 1, change directions, K is equal to 1. That gives you your T equals 3 structure. The main part of the structure here is a beta barrel. This should look really similar to another kind of positive strand, single-stranded RNA virus with a pseudo T equals 3 structure that we've been reviewing for our exam. Right, the picoRNA viruses. So polio in particular has a very similar structure here. This beta barrel structure here, it's actually a little bit different relative to what's going on in polio. All of these viruses and the CucumberMosaic virus and the close relatives have this very arginine-rich N-terminal alpha helix. What do you think this is interacting with? So arginine is what? One of those few amino acids I expect you to remember. It's positively charged. What's it going to be interacting with? Negatively charged genome, RNA. So that's on the inside. So immediately this tells you that this is on the outside and this is on the inside of that. So how do these, again, pseudo-equivalent T equals 3 icosahedral particles get inside the cell? The way they do it is getting some help. So almost all plant viruses, with a few little exceptions, are vectored, i.e. they've got some other mechanism for getting inside, not even so much the organism, but even inside the cell. Most plants have these really thick cell walls. It's really tough for a virus to get through these things. Plants don't usually have all of these extracellular proteins that are going to be communicating with the inside. Plants don't do receptor-mediated endocytosis. So how do you get a virion inside a plant cell? Basically, it's by chewing through them. And this is the aphid, which is doing that chewing right here. One of the, I think, really fascinating aspect of CucumberMosaic virus and a lot of related viruses is that to get a productive infection, you need multiple genomes and multiple virions. And that's because each of the RNA segments, which is part of your genome, is packaged in a different capsid. So different virions. And this seems to be a really inefficient way of packaging your genome, because if just one virion gets in with one RNA, that's not going to be enough. A different virion with a different RNA gets in, that's not going to be enough. You've got to get all of them together. And the reason that seems to work is because, yeah, this is an electron micrograph of an aphid, but it's still ginormous relative to the number of virions which are there. You need the TEM. And so the mouth parts of this aphid, and in fact a lot of the plant-eating insects, is just absolutely covered in virion particles. You can literally scrape virions off of the mouth parts of these things and look at them in the EM. So very large inocula you're getting there. So if you're thinking about transmission, aerosols, the kinds of things we've been talking about with a lot of these human viruses, just you're only able to get a couple of virions in any of those individual aerosols. So doing this split capsid kind of thing is not going to be terribly successful. On the other hand, if you're getting into the cell through one of this real damage processes, then you can get a bunch of virus particles in at the same time. Most plant virologists don't use aphids. They actually literally just scratch the top of the leaf and then put their virus on top of it. works just as well. So physical damage. It seems to be basically physical damage. Making a hole through the hole is how the virions get in. Yeah, there's a question?
Doesn't the virus have to replicate in the vector at least a little bit in order to be able to be passed on to the host?
So the question is, does the virus need to be able to replicate here in order to get passed on? The answer for most of these plant viruses seems to be no. It doesn't need to replicate, partly because many of the virions are very stable, so they can hang out for a pretty long time, and partly because usually it doesn't take very long for an aphid to get from one leaf to the next leaf, or even from one plant to the next plant.
They do in mosquitoes, right, as a vector?
Right. So the mosquitoes usually, so the question is, so for mosquitoes, for a lot of the animal viruses, those do end up replicating inside the mosquito. It's partly because what's happening with the mosquito is it's taking blood in, and then the blood is coming back out, or it's also the salivary gland. So it's actually got to move through the actual mosquito. Whereas in this case, it's all on this messy eater part of the aphid. So it doesn't seem that in most cases you need replication going on here. And there's enough. One of the things that you do find, and one of the I was thinking about picking up a cigarette butt on my way over here. If you just look at those and take a tobacco leaf and scrape it and put some cigarette butt on it, you'll get infection with tobacco mosaic virus. So it gives you an idea of how stable some of these things are. So what's in these capsids? Again, there are multiple capsids that you need. In the case of cucumber mosaic virus, one of these icosahedral particles has the 1A RNA in it, or the RNA1, excuse me. One has RNA2, and one has RNA3 and 4. And you need all three of these to infect one cell in order to get productive replication, which is still, I think, pretty amazing that evolutionarily this is what works. But again, it's because it's hard to get through the cell wall of these plants, and you've got high copy numbers of the virus, it will get in. So you're going to end up with, again, this is a Poisson distribution as well. So if you've got really low multiplicity of infection, it's never going to work. You're never going to have three, and particularly three different capsids with the three different RNAs in them. So you need to have really high inocula in order to get productive infection. If you look at these RNAs that are present in the individual capsids, they've all got 5' caps on them. What does that tell you immediately about some of the proteins that are encoded there? They encode capping enzymes, exactly. So the cap is not being stolen in this case, which we had for influenza virus. These all come in with caps that are made by the viral proteins. They also all have, at least in the case of the cucumber mosaic virus, and it's true for many other viruses, plant viruses as well, but particularly for the cucumber mosaic viruses, they have these really interesting structures at the 3' end of the genome that's here shown as a clover leaf. And clover leaves immediately make you think of tRNAs. So tRNA-like, well, if I look at this, it doesn't look to me much like a tRNA. But I haven't seen, if you look at the three-dimensional structure of some of these things, they actually do look quite like tRNAs, which of course don't look like clover leaves at all. They're much more like L structures. So this is what the 3' end of each of the segments of the cucumber mosaic virus genome looks like. So here's the very 3' end right here. That then, if you just follow, and it took me a while to follow this whole thing through. So here's your 3' end that base pairs a little bit with itself, and then it goes over here, base pairs with itself again, all the way around here, then makes a loop back to here, and then you have the rest of the genome. So it's this really convoluted secondary structure present at the end of each one of these segments. So, yes, tRNA-like. How do we know that it's tRNA-like, even if you didn't have a structure for it? It's because the CCA enzyme, which puts a CCA tail at the end of all of your tRNAs, and that's where you're going to get the attachment of your amino acid by the amino-acyl tRNA synthetases, also acts on these RNAs. So you have the CCA enzyme that binds to them, and in some cases, actually will even add an adenine here at the very 3' end of the genome. The other thing is that it looks so similar to a tRNA that aminoacyl tRNA synthetases. We'll actually put amino acids onto this 3' end. Why? It's not because the genes are actually coding for things that have amino acids at the end of them, but it's probably just like what you get with poly A tails. What's one of the reasons you have a poly A tail? Protection. Protecting the end of the genome from the nasty exonucleases, which would just love to chew up the ends of your genomes, and that's what it does when it's degrading messenger RNAs in general inside the cell. So that's what this structure seems to be involved in, is sometimes adding to the genome, but mostly getting protection for the rest of the genome. What are the proteins which are made here? The important ones are, again, not surprisingly, a capping enzyme. Here, it's RNA1, methyltransferase, and then all of these other RNA viruses also have what? RNA-dependent RNA polymerases. So you have these. What else do you need? You remember back to way back when, MS2, Q beta. Got to make your virion, right? What else do you need to encode? You need to encode a protein. So coat protein is this RNA4 here. So that's all you really need. Something to cap, something to replicate, and a coat protein. So there are other proteins which are encoded here. The main thing has to do with movement, getting from one cell to the next. And so there are some what are called classic movement proteins. This RNA3 basically encodes one of those, and this other RNA4 does it as well. So movement proteins, capsid proteins, RNA polymerase, and capping. Where does this, sorry?
You want us to know like the protein or the RNA?
In this case, well, they're, 1A is RNA1, 2 is 2A. I can't remember that it's 2B that's an RNA4A, and particularly in this case, I hate this nomenclature, and it's 1A, 2A, 2B. Okay, it just happens to be the structures that you have there. I would know what the activities are, but not necessarily the numbers. Again, I'm not going to know this till I write the exam, which will be late Tuesday night. As usual, right? So it'll be good timing. So where does replication take place? There are the regular viral proteins. Again, are RNA polymerase and proteins that are involved here in capping. These we're going to bind to your RNA. Again, it's got this 3' tRNA-like structure. One of the things that seems to happen with most of these single-stranded RNA viruses in plants is that they hide themselves to replicate. Why are they hiding themselves to replicate? Probably because cells, and particularly plant cells, are really good at getting rid of double-stranded RNA. In fact, RNA interference was originally discovered in plants, where you had double-stranded RNA, which would then suppress expression of different genes. So probably the reason that you have these special structures which get made during replication is probably just to protect this double-stranded RNA intermediate from getting degraded. There are another couple of things that you have to have in order to get normal replication here. Lots of translation proteins. This should sound really familiar and similar to which RNA virus that we've talked about so far? Not that I'm specifically testing on this midterm. Give you a bit of an idea. The one that I had just mentioned in terms of what minimal numbers of proteins you need. MS2, so the RNA bacteriophage. They also need translation proteins, cellular translation proteins, in order to get good replication and packaging to take place. So, number of these cellular proteins, again, particularly the translation proteins. EF3 is one of the initiation factors. And then there's a DNA J-like protein which is required. This is a protein folding enzyme, probably for assembly of the individual virus particles. So those are, again, not terribly different. What is different as far as these plant viruses is, again, how they get around. So movement protein, in the case of CucumberMosaic virus, is 3A. Other plant viruses also have movement proteins. And the best studied of these movement proteins is actually the TMV movement protein. But anytime you go to a virology meeting and people are talking about plant viruses, they're all about movement protein this, movement protein that, movement protein this. Because it's actually kind of hard to get around plant cells. Again, because they're pretty tough and they can't run away. So they want to be very careful about controlling the transmission of different things between different cells. So the plasmodesmata are the classic case of connection from cell to cell, because the plant does need to communicate between different cells. These plasmodesmata are really small and usually only allow very small things through them. These movement proteins, at least some of them, cause an expansion of the plasmodesmata so that RNAs, and particularly RNAs together with RNA-dependent RNA polymerases, can get through them. Apparently, full virions can't get through them. But this is your local transmission, plasmodesmata. You may remember back here, you read the table carefully, cell-to-cell movement. That's what's happening with 3A. Long-distance movement here is getting from these cells, which are the ones which are getting chewed up by the aphid, through to the rest of the plant. And that's the whole plant vascular system, xylem and phloem here. Most of you probably know way more about this than I do. But these are the cells that virus or the virus RNA needs to get into to be transported through the rest of the cells. So there are really two kinds of movement: cell-to-cell movement and then long-distance movement that have to be taking place here. There's one other thing which I forgot to mention about these proteins. Because you've got such a strong double-stranded RNA response, usually in plants, once they get infected by these plant viruses, there's also, not surprisingly, a plant virus protein, which is combating this. So again, if we go back to our table, this 2B protein suppresses RNA silencing. And this has been one of the big areas of research in a lot of these plant viruses, because RNA interference is a really big deal. Got a Nobel Prize a couple of years ago. Double-stranded RNA leading to silencing, cleavage of different RNA products. And so how plant viruses are dealing with this, how they've been antagonists to this process, has been a really active area of research, and in fact a lot of it down at Oregon State. They were the first people to in fact discover this particular effect that was going on there. So that's our CucumberMosaic virus, all the chapter in about half an hour. Again, very, very wide host range in terms of what it can infect. Lots of different kinds of plants. The structure, nice, easy, boring, beautiful. Entry and transmission, it's all about making damage to the cell wall of the plant. Once it gets in, the movement proteins help it move to the plasmodesmata, but also get into the plant vascular system for moving things around. Any of you work on non-vascular plants? I think it's a really fascinating area for looking for viruses. Anybody looking for a great research project? Viruses of non-vascular plants. Yeah, in the back.
I just wonder, where's that vesicle, that budding happening that you showed?
Oh, the budding which is happening when you're getting replication, that's inside the cells. So, and it seems to be associated with the endoplasmic reticulum. There's, kind of a dirty secret of talking about this, is a lot of this work was done in yeast, which is not usually the host for these particular viruses. But as true with most positive strand RNAs, once you put it in, it will still replicate and translate. So if you can get all 3 of those RNAs into a plant cell, and you don't need the coat protein because it's not going to be spreading. But if you're looking at replication, and of course there are wonderful genetic tools, great cell biology tools for looking at this in yeast. It's probably happening in plants too. It's just a lot harder to study those individual plant cells. So probably the endoplasmic reticulum in the plants, in an individual cell. Yeah?
So when the plant's making the virus, the virus just goes somewhere and hangs out until the plant gets eaten by something.
So yeah, how does maybe I'll paraphrase your question is how do viruses get spread from plant to plant?
Right.
Yeah.
So it just like builds up the quantity of virus and then just like waits to be eaten.
Right. Waits. Yeah. So again, waiting. This is again a little bit of an over-anthropomorphization here. But the real question is again, how do you get transfer? How do you get transfer from one plant to the next? An uninfected plant gets infected from an infected plant, and it does basically seem to be, as you're saying, waiting for some insect to feed on that plant and then transfer it to a different plant. So that seems to be the mechanism of getting transport taking place there. There are some examples of the virions being infective through roots. That's kind of a controversial issue here. But it seems to be in most cases you get very high amounts of virus production in these plants, humongous numbers, because that's what you have to have. Yeah, sure, the aphid's pretty big, but it's not going to be eating that much of the plant. And so you've got to have very high titers wherever that aphid happens to be eating in order to get transfer to take place.
Where does the virus build up?
It seems to build up in most cells. So many of the cells, particularly in leaves, is actively growing. And we'll get back to that when we talk about tobacco mosaic virus a little bit later on. But actively growing parts of the plant. So the leaves, the stuff that look yummy.
Where inside the cell does it like store or whatever?
Oh, so where do you get an accumulation of virions? So if you go back, sorry to go back here real quick, but your plant cells, most of them are filled with these vacuoles. And the cytoplasm is generally relatively small for a lot of these plant cells. If you look at a virus-infected plant, most of the cytoplasm will have virions in it. So it's pretty much in the virions, so just basically right underneath the cell wall. So as soon as that aphid comes in, takes a little snack, then it's got virions associated with it. But the numbers have to be really high to be able to do this and actually get proper transfer, because you need 3 of the virions in order to get this active infection. So it's really, again, it still blows me away that this has been evolutionarily so successful. It works. Okay, so I want to talk about a few other things here, some of the other viruses as well. One of the most fascinating aspects of a lot of cucumber mosaic virus infections is that many of them have these satellite RNAs which are associated with them. This is the whole sequence of some of those satellite RNAs. So how the heck could something like this actually have some kind of function in terms of disease? What's going on there? These were actually discovered way before people got really excited about small RNAs being regulatory and interacting with all kinds of other things inside cells. The plant virologists had known about these for decades. Oh yeah, sure, small RNAs are really important. They cause differences in terms of disease. How they cause differences, nobody really knows. But these short RNAs are really quite common. You've got some short RNAs that seem to cause a lower amount of disease, some which seem to exacerbate disease. Quite how that's happening. These RNAs are presumably also picked up through the capsids, so they just get encapsulated. They're replicated by the viral RNA-dependent RNA polymerase. So you get lots of these. Turns out that these 3 residues down here at this end of this particular RNA, which leads to a nasty phenotype, much worse disease. If you change these nucleotides, then basically all disease goes away. Again, very open question about how these things are actually functioning here. So lots of questions that people are still trying to answer. So I want to talk a little bit about CCMV, cowpea chlorotic mosaic virus. because this virus is, like a lot of these virions, very, very stable. And it kind of has to be stable if it's floating around on aphids, and it's got to be hanging out inside the cytoplasm of your plant cell in order to be able to get transferred later. But if you got a really tough virion, how does it let its genome out, or its segment of the genome? So what happens in this case is, again, it's just like the kid's toy. It's compacted on some cases, but as soon as you change the pH or the ionic strength, which is going to be different outside of your aphid or inside the plant cell, it opens up. And this is a completely reversible process. And it turns out you can just express the capsid protein from CCMV, and it will make this particle, which will undergo this reversible process. So really amazing nano-sized process. These are about 25 nanometers in diameter in terms of going back and forth here. Normally what would happen is the virus genome is going to be, or should say the genome segment, comes out here. In the case of CCMV, it's actually 2 segments rather than 3. And then will be translated. You'll make more virus particles. But a couple of very smart people, way smarter than me, including one of my postdoctoral advisors, said, well, if RNA can be coming in and out of here, maybe you can put other things in and out of there. And the first one they thought of was metals. Now, why metals? Metals are, of course, very interesting for all kinds of different things, but in this particular case, what they were looking at were ferromagnetic metals. And one of their main sponsors for this project was Toshiba, who were really interested in how do you raise the capacity of hard drives, because what is on a hard drive is a bunch of magnetic particles. And it really depends on how small you can make those magnetic particles. Well, it turns out you can take CCMV and put magnetic particles inside them. You open them up, put the magnetic particles inside, put them back together, and then because you can get really ridiculously large numbers of virus particles, virions, then lay these down on basically, again, what would be a hard drive, burn off all of the capsid proteins, and you've got these perfectly spherical 25 nanometer magnetic particles. So basically raises the density of magnetic storage by about 2 orders of magnitude, just by using these virus capsid shells. They're also very interested in using these for magnetic resonance imaging. If you can engineer the structure of the protein on the outside so that they interact with, say, a cancer cell, you put a contrast agent on the inside. This could be really, really useful for MRI. In fact, they've patented some of this, and some of this I think is even already in the clinic. Some chemotherapy drugs, same idea here. You can put RNA in and out. You can put whatever else you want in and out. You got this reversible condition. Chemotherapy drugs, lots of ideas about how this is happening. Again, Mark Young is the guy I was working with at Montana State University, although I wasn't working on this project at all. And Trevor Douglas, who was at Montana State, has now moved to Indiana University. Interested in some more applications here. Particularly Trevor's doing some really cool stuff as far as this is concerned. Mentioned hot viruses other than the ones we're working with in my lab. But this is one of the plants that you see when we are literally going out into the field, into the hot springs, and looking for various different viruses. It's a plant called Dicanthelium lanugnosum, also known as Texas panic grass. I much prefer that. Really wide-leafed grass that you find at ridiculously high temperatures. This is not Fahrenheit, this is Celsius. So these plants can grow perfectly happily up to a little over 50 degrees Celsius, which is pretty darn amazing. And in fact, we can go out in the field when we're in some of these hot spring areas, Yellowstone, Lassen Volcanic National Park, and just by seeing where the panic grass is, tells you what the temperature of the ground is. it's really pretty amazing. We can stick our thermometers in there, you get 50 degrees. One thing that was also being done at Montana State when I was there, it turns out that this grass can only survive at those really high temperatures when it's infected with an endophytic fungus. And so this is the endophytic fungus right here, Curvularia protuberata, found in lots of grasses, but this particular one is found in that dicanthelium. But it's not just only when they're infected with fungus. What kind of class? What's the name of this class? It's a virology class. So it turns out that it only gets thermal tolerance if the fungus is also infected with a virus. And so these are the data down at the bottom here. So normal, infected with fungus and virus. Here, this is without the fungus. This is, but, and here is without the virus. So these guys are perfectly happy at 50 degrees. This one has no fungus, but this one has fungus and no virus. So you have to have fungal infection and virus infection in order to get this thermal tolerance. So this is a cartoon, basically, just of how this works. So here's our dicanthelium when it's got happily infected with fungus and with this Curvularia thermal tolerance virus, CHTV here. On the other hand, they're not colonized. They're really unhappy. There's a nice review article that Marilyn Rusnick wrote. In fact, the same author as the chapter here on this CucumberMosaic virus. So this is one of my favorite viruses that I don't work with because it's giving thermal tolerance. How it does this, there are a number of theories, but nobody really knows what's going on there. So it's not really a plant virus, but close enough. Real plant virus, on the other hand, tobacco mosaic virus. So this is the original Contagium vivum fluvium. This was found by Ivanovsky and Beijerinck, probably independently, but from each other. And the data are really pretty good on that. No one was reading the Russian in one case, and nobody was reading the German in the other case. I just pulled up this particular example here. Tobacco mosaic virus. Anybody know what this plant is? Is it tobacco? It's tomato, exactly. So tobacco mosaic virus actually infects tomato also really well. So last night, digging through the internet, pulled up Beijerinck's paper from 1898, Martinus, Willem, Beijerinck, and I just love this quote. I'll let you guys read it. I wish I could use these things. Yeah. Incontestable experiments. I would just love to have an incontestable experiment that I could publish, or even to write any of this thing up here. This is the link, in fact, to the whole thing if you want to take a look at it. But I read this paper. It was really amazing, the stuff that he was already showing in 1898. He uses virus as well, so it's not just Contagium vividum fluvium. Shows, at least has some data, that it's only able to replicate in growing plant tissue. So he immediately, or say not immediately, but makes that connection that the virus can only replicate in something else that is already growing. And he shows heat stability. So he did experiments going up to 40, 50 degrees Celsius and drying out the virus. It was still infectious. So again, 1898, he's already making a number of predictions about how this is working. They had the diffusion experiment, which is probably incorrect, but just really amazing that that was what was happening there. So first virus as well, that in and of itself is probably not enough to write a book about. But people got really excited about tobacco mosaic virus, mostly because it was this really simple structure and something that people could work with. And again, Contagium vivum fluvium, vivum, alive, but a particle. So we've got these alive particles. Is it really alive? Is it a particle? What's going on here? So one of the major people who was working on this, Wendell Stanley, got the Nobel Prize in 1948, basically for crystallizing TMV. They got the first crystals in 1935. Wendell Stanley was at the Rockefeller Institute, got his Nobel Prize, and Berkeley said, hey, we need a Nobel laureate. We'll hire him. And then we'll name this building after him. And so this is, or say this was Stanley Hall at Berkeley. And this window right up here in the corner, right behind that is where I did one of my rotations for my PhD, in this building. Giving you a bit of an idea, this building doesn't look like this anymore. Now it looks like this. It was completely rebuilt in 2007. Again, nothing else is left here. This is the new Stanley Hall down at Berkeley. It also tells you that they have a little bit more money than we do for putting together some of these things. Oops, it's one too far. Eek! Uh-oh, I was encountered a problem. Okay, fine. No, I don't want to send anything here. Let's see if we can restart it here. That's what happens when I edit. You were talking about me editing my lectures from year to year. Obviously, this was a problem. So I should never change a running machine or whatever we should say here. So where were we? So this is the structure that was not originally found, much later found after Stanley was able to crystallize the virus. Let's look at it here. What's going to work? Yes. Okay, good. So this we've actually looked at before. This is the really classic helical structure, tobacco mosaic virus. All of these proteins bound on the outside of the RNA. This is a naked virion, so the nucleocapsid doesn't have an envelope around the outside. The protein itself is here. It's basically a 4-helix bundle with this loop at the end here. I'll give you one guess what kinds of amino acids you have in this loop. These are going to be the positively charged amino acids, which then hook up into the RNA here. I'm going to try and go to this link, and everything may crash abysmally, so we'll see what happens. This is a link to the protein database and the tobacco mosaic virus. Again, this is where I got that structure from. This is a structure now looking at just 3 of those individual rings, and we're going to see if we can get this to work in JMOL. With any luck, it will come up. You can now use this and turn the structure around, zoom in, zoom out, rotate. What I'm doing is take a look at the side here. So there you can see really nicely how each of these rings is slightly too big to go all the way around, and that's what's giving you the structure as it's wrapping around this RNA and packaging the RNA all the way up through the top here. I can spend hours playing around with these things. But that's that loop here at the end, which is sort of looping around the RNA, giving a little groove in which the RNA is being packaged. And if we zoom in a little bit here, no, that's the other way to go. Not zoom out. Zooming all the way in. Okay, now we can't see it anymore. It's all gone. Okay. My computer's just too old here. We can spin it around. But what I wanted to show was those helices, the 4 alpha helices, which are sticking together, and then each of those are all stacking on top of each other in terms of the rest of the helix. We could talk about pandemic flu for 10 minutes, or we could go outside in the sun and pretend it's the weekend. Good. Okay. See you on Monday.